dear students, today we will learn about the extra-embryonic membrane or the fetal membrane. In our last class, we have learned about the structure of placenta and the function of placenta. And we learned that how does the placenta act as endocrine glands. So today we will learn about the extra-embryonic membrane or the fetal membrane. So what are these membranes actually? So actually this extra-embryonic membrane, they provide protection and nourishment to the developing fetus. That means this extra-embryonic membrane, they are very essential for providing the nutrition or nourishment and protections to the developing embryo. Number two, they are mainly originated from the trophoectoderm or from the trophoblast. If you remember the structure that we have learned, just try to recall the structure of blastocyst. When we are learning about the blastocyst, the outermost layer, alright, the outermost layer was known as the trophoblast. So look over here. So if we are trying to draw the structure of blastocyst, let us say, so this outer cell, the structure was known as the trophoblast or the trophoectoderm. Then, in the inner side, you'll find the embryonal axis, where you find the cell known as inner cell mass. The cell of the inner cell mass will give rise to the embryo. Clear? Whereas the trophoblast does not take part in the formation of embryo, but they are essential for the formation of extra-embryonic membrane. So all the structures we have learned in our previous class, try to recall it. Clear? So the outer cellular structure is known as the trophoblast, and from this trophoblast there will be formation, there will be origin of extra-embryonic membrane or the fetal membrane. Alright? Now, what are the types of extra-embryonic membrane? Clear? So generally, the different types of extra-embryonic membrane are, we can note it down, amnion, chorion, allantois, and yolk sac. So these are the different types of extra-embryonic membrane which are developed or which are originated from the trophoblast or trophoectoderm. Clear? Now we are going to look at the structure of this extra-embryonic membrane. So look at the figure. So from the textbook, I have the figure number 3.26. So let's see the different structures. Alright. So this is the embryo part, as you can see over here in the figure. This is the embryo part. Now, close to the embryo, you can see a sac-like structure, SAC. Okay. Close to the embryo, you can see the origin of a sac-like structure. So these particular structures which you can see over here. This structure, this sac-like structure is called the yolk sac. Okay, what is called? It is the yolk sac. Clear? And similarly over here you can see the second sac-like structure which is called the allantois. Yes, these are the sac-like structures which are close to the embryo. So we are finding the yolk sac and the second sac-like structures, these structures which is called the allantois. Clear? Now, the entire embryo, look over here, the entire embryo is surrounded by a thin layer. Can you see this layer? We are talking about this layer. Okay, we are talking about this layer. The entire structure is surrounded by a thin layer. So the entire embryo is covered by a thin layer, and this layer is known as amnion. What is known as? It is known as amnion. So amnion is a thin layer that completely surrounds the embryo. And over here, in this particular cavity, we will find the fluid which is called the amniotic cavity. Okay, so this particular cavity is called amniotic cavity, and this amniotic cavity, it contains a fluid which is called the amniotic fluid, which we will discuss afterwards. Clear? and the last membrane. You can see over here, the entire structure is surrounded by this last autonomous layer, which is called the chorion. C-H-O-R-I-O-N. Chorion. Clear? And these are the chorionic villi. So these are the different parts or the different structures of the extra embryonic membrane or the fetal membrane. So this is the embryo part, this is the yolk sac, this is the embryos. The embryo is covered by a thin transparent layer, which is called the amnion, and the entire structure is protected by a chorion. And we will find the chorionic villi over here, which will give rise to the placenta. Clear? So now we will discuss one by one. Clear? So this is the one of the easiest part we can say in this topic. We are not going to go in details. Clear? Just simply toss it. We will describe the structures and their functions. So first one, let us talk about the amnion and the amniotic cavity. Now before going into the amnion and the amniotic cavity, we'll just try to recall the structure of the gastrula, alright, during gastrulations. Now you can see over here, we have learned that during gastrulations, mainly find two types of cells. The embryonal axis will give rise to embryonal knot. Clear? Embryonal knot will give rise to embryonal axis. And over there, we will find two layers of cells called the epiblast cells, the columnar cells, and the hypoblast cells. Remember, we will find the two layers of epiblast cells and the hypoblast cells. This we have learned while learning about the gastrulation. So here we have got the epiblast cells and the hypoblast cells. Now during the process of gastrulation, the two layers of epiblast, let's look over here. Outer layer and the inner layer, these are the two cells, first cells, second cells of epiblast. So these two layers will separate. So you look in second figure, the two layers are being separated. So a cavity will be created in between the two layers of epiblast cells, and this particular cavity is called the amniotic cavity. Clear? And the roof of this amnion, alright, the roof of the amniotic cavity, which goes with the ectoderm, is called the amnion. So now we are going to learn about these structures, amnion, and we are going to learn about this amniotic cavity. Clear? So let's see what is amnion, and what is amniotic cavity, and what is their functions. So amnion is a thin protective membrane around the embryo. If we are trying to recall it back, in the structure we have learned that this particular layer that completely surrounds the embryo is the amnion. Clear? So amnion it completely surrounds the embryo. Now it develops from the ectoderm lined by mesoderm. Now what do you mean by ectoderm and mesoderm? Right, these are the embryonic germ layer cells. We have learned during gastrulation that there will be formation of three embryonic germ layer: ectoderm, endoderm, and mesoderm. So ectoderm, mesoderm are the embryonic germ layer cells that will give rise to different organs and different tissues in the developing fetus. Okay? So this amnion, it develops from the ectoderm and is lined by the mesoderm. Clear? Number 3 point is very important. The amniotic layer or the amnion is somatoplure. Now what does somatoplure mean? Somatoplure means that its inner part. Alright, in the inner part it is composed of ectoderm, and in the outer part is composed of mesoderm. So remember this one, somatoplure. In the inner part we will find the ectoderm, and in the outer part we will find the mesoderm. The space between the two layers of the epiblast develops into amniotic cavity. That means these two layers, alright? These two cells of the epiblast, they will separate during gastrulations, and a cavity will be created, and this cavity is called amniotic cavity. Now in the amniotic cavity, we will find the amniotic fluids. Clear? So over here, so in this particular cavity, this is the amniotic cavity. Alright? And in this particular cavity, we will find a fluid which will provide the aqueous medium to the developing embryo. So this particular fluid, the amniotic fluid, is mainly formed or secreted from the fetal part and from the maternal part, and it provides the aqueous medium to the developing embryo. Here, next one. The roof of the amniotic cavity forms the amnion. One more time. The roof of the amniotic cavity forms the amnion. So over here. So this is the amniotic cavity, and this particular roof of the amniotic cavity is called the amnion. Clear? It's clear for you? So, amnion is a thin fluid membrane that surrounds the embryo, that covers the embryo. It develops from the ectoderm, it is lined by mesoderm. The layer is somatoplure. In the inner part, we will find the ectoderm, and in the outer part, we will find the mesoderm. The space between the two epiblast cells forms a cavity which is called the amniotic cavity, and it contains a fluid called amniotic fluids, and the roof of this amniotic cavity forms the amnion. Now, what is the significance of amniotic fluid? That's very, very important. Okay, so we are going to learn about the amniotic cavity functions. Then we will also learn about the amniotic fluids also. So, amniotic cavity function, it serves as a shock absorber for the fetus. That means it provides protection to the developing fetus. It regulates the fetal body temperature. It prevents the desiccation of the fetus and prevents adhesion between the fetal skin and the surrounding maternal tissue. So these are the general functions of the amniotic cavity. Now, what is the function of amniotic fluids? I told you that it provides the aqueous medium for the developing embryo or for the developing fetus. Not only that, since the amniotic fluid contains the cells from the fetal part also. Listen carefully. The amniotic fluid contains the cells of the developing fetus. Now that particular cells which are present in the amniotic fluids are utilized to detect whether there is any type of disorder in the developing fetus or not. Clear? That particular technique is called Neocentosis, and that particular technique is also utilized for the process of sex determination. But owing to the increasing female fetus death, you can say, this process has been banned. It's illegal to perform amniocentesis because we have learned about that if the developing fetus will be female, then that particular fetus will be killed. Right? So therefore, this amniocentesis has been totally banned. But it was very essential to detect the particular type of genetic disorder in the developing fetus. Genetic disorder means which can be inherited from the parents to the offsprings. So those fluids, amniotic fluids, was essential for the detection of those genetic disorder in the developing fetus. Clear? But at the same time, it was also essential or it was useful in finding out the sex of the developing fetus. Clear? So therefore, this is the function of amniotic cavity, and we have just discussed the function of amniotic fluids. Now next one, yolk sac. The yolk sac, it acts as the embryonic gut. Look over here. It acts as the embryonic gut. That means it will store the food particles. Clear? It stores food in the form of yolk. It will store the food, and those food will be in the form of the yolk. But in case of human beings, these yolk sacs are poorly developed. Clear? These yolk sacs are poorly developed, and in case of development, it usually decreases. Clear? Why does the yolk sac in human being is not well developed? Answer is that because in case of human being, the embryo or the fetus will get its nourishment from the placenta. Clear? So there is no need of the yolk sac. So that's why it is poorly developed, because the fetus or the embryo will obtain its nourishment from the placenta, and also it obtains the nourishment from the endometrium of the uterus. So owing to the presence of placenta, and since placenta provides the nutrients, nourishment for the developing fetus, this yolk sac is poorly developed in case of human being. Clear? Next one. In second and third week, yolk sac transfer the nutrients to the embryo, and during the fourth week, it forms a part of the gut. So that's why it is called the embryonic gut. Clear? So we'll not go in details. Then this next part, let's see. Now, how does it will be formed? The cells of the hypoblast migrate along the wall of the blastocyst cavity and forms an extracellular membrane. Now, you might get confused over here which part is hypoblast and which part is blastocyst cavity. Okay, so we'll just go back to the structures and we'll see the cells of hypoblast and the blastocyst cavity. So look over here. This is the blastocyst cavity, and this is the hypoblast cell. These two are the blast cells that will help in the formation of amniotic cavity and the amnion. Now this particular hypoblast, it will move towards the cells of blastocyst cavity and forms the yolk sac. So this is the hypoblast, this is the blastocyst cavity. Clear? And that these two are essential for the formation of yolk sac. Now, next one. So the cells of the hypoblast migrate along the wall of the blastocysts and form the extracellular membrane. Now, extracellular membrane together with hypoblast, they will form the yolk sac. Clear? So, hypoblast cells along with the extracellular membrane, they will form the yolk sac, and yolk sac particular layer is Somatoplure. Meaning is that in the inner part you will find the endoderm, and in the outer part you will find the mesoderm. One more time, the yolk sac layer is Somatoplure. In the inner part you will find the endoderm, and in the outer part you will find the mesoderm. Now you can get confused on which part are you talking about. Alright, the inner part and the outer part. If you look over here in the given figure, the yolk sac. Now this yolk sac is mainly surrounded by a second layer like this. Clear? So this will be the outer membrane or the outer layer, and this will be the inner layer. Clear? I have this clear for you now. So this particular layer is Somatoplure. In the inner part we will find which layer? We will find the endoderm, and the outer part we will find the mesoderm. Now next layer, allantois, the second sac-like structure. So we are going to talk about. Okay, it acts as the embryonic kidney. Remember, yolk sac, it acts as the embryonic gut, whereas allantois, it acts as the extra-embryonic kidney. Now, it stores and removes the nitrogenous waste products. So whatever the waste products will be generated in developing fetus, alright, it will be stored and removed by that waste product. But over here also, it is poorly developed in case of humans. Why? Why the allantois is poorly developed in case of humans? Simple answer. In case of human beings also, the nitrogenous waste products are simply removed by the help of what? Placenta. Yeah. Now since placenta will simply help in the removal of waste products from the developing fetus, so this particular allantois is also poorly developed, because placenta is mainly acts as the organ for excretion also. So whatever the waste products will be generated by the fetus, it will be removed by the help of placenta. So this allantois is also poorly developed in case of human beings. Next one, the layer is Somatoplure. In the inner part we will find the endoderm, and in the outer part we will find the mesoderm. So kindly remember the words. The layer can be Somatoplure or it can be Somatoplure. Clear? Remember these things. Now, chorion, the last extra-embryonic membrane, chorion, which usually surrounds the entire part. Right? If you remember, which part we are talking about? We are going to talk about the last membrane, which entirely surrounds the structure of the embryo. This part, outermost layer, chorion. Clear? So we are going to talk about this layer. These are the chorionic villi, and this particular layer we are going to talk about. Generally, what we find is that You can see the structures. We are finding the embryo part. We are not going to draw it. Okay, we will draw the embryo part. And when we draw the embryo part, we are finding the embryo over here. Clear? Then we will find the yolk sector like this. Yolk sac and allantois. Now this entire structure is surrounded by the third layer. So I'll just simply draw it like this. Alright, so we are talking about this layer. This layer is called the chorion. And over here we will find the chorionic villi. Clear? So now we are going to talk about this outermost layer, which is called the chorion. And this one, if you are seeing over here, is the chorionic villi. Clear? So now we will just simply talk about the last layer, which is called the chorion. Is this clear for you? Alright. So let's see what is chorion now. So can you hold it down? Chorion, it completely surrounds the developing embryo. We have seen structures where the chorion was surrounding the entire developing embryo or the developing fetus. Now it is the principal embryonic membrane, it is the most important embryonic membrane. It forms the portion of placenta and provides nutrition to the developing embryo. What to talk for you? Well, let's see next part. Functions. The chorion protects the embryo and the fetus from the immune response of mother. But how? How does the chorion protect the embryo and the fetus from the immune response of mother? You see the answer? Answer is there. It secretes, the chorion secretes proteins that block antibody production by the mother. Clear? So you know what antibody? Antibody are produced in response to the antigen. Clear? So this particular chorion, it produces the proteins which will block the antibody production by the mother. Number 2, promoting the production of T lymphocytes. Now what is T lymphocytes? It's a type of WBC. There are 2 types of WBC or lymphocytes, T lymphocytes and B lymphocytes. So it promotes the production of T lymphocytes that suppress the normal immune response in the uterus. Suppress means to inhibit. It will decrease the immune response in the uterus. And number 3, it also produces human chorionic gonadotropin hormone. So these are the functions of chorion. So till now what we have learned? So now we have learned about the extraembryonic membrane or the fetal membrane. We have discussed about the 4 particular membranes. First one was amnion, where we discussed about the amniotic cavity and the amniotic fluids. Number 2, yolk cell. Number 3, allantois, and lastly was the chorion. Clear? So kindly go through it. Now simply we discussed about the difference between embryo and the fetus. Now what is the difference between embryo and fetus? What is embryo and what is fetus? Clear? So embryo means over here, it develops mainly from the time of fertilization. Clear? So when the egg and the sperms will be fused, there will be formation of a zygote, that is single cells, right? And that cell undergoes cleavage, leading to the formation of morula, blastula, blastocysts, and goes for implantation. So that particular time period is usually called the embryo. Okay, so let's see over here. So developing organisms from fertilization to the end of the 8th week of development until all the organs are differentiated is called the embryo. That means from the time of fertilization till the development of organisms. Till this time there is no such organ differentiation. The organ has not been differentiated. Simply that particular cell zygote is under the process of development. It is developing into morula, blastula, gastrula. Clear? Then it undergoes for implantation. Then we call it the fetus. It is ongoing developing organisms of viviparous animals. Now what are viviparous animals? That give birth to the young ones. Since the most of the attachment with the uterus, that means when that blastocyst goes for implantation, then we will call it the fetus. Now human embryo from the 3rd month onwards will be known as the fetus. So from the time of fertilization till the time of organ differentiation, we call it embryo. After implantation, alright, after implantation, when there will be the process of organ differentiation, process of gastrulation, process of formation of 3 embryonic germ layers, then that particular one will be known as the fetus. So human embryo from the 3rd month onwards will be known as the fetus till the birth. Alright, from 3rd month onwards till birth is known as the fetus. Clear? So kindly go through it, go through the diagram. take the notes and please keep on studying. Clear? So in next class we'll talk about parturition and we'll talk about the lactation.